lesson. Today we're going to be focusing on Mendelian genetics. My name is Kelly Hollis and I'm a biology teacher, and today we're going to focus on a number of syllabus dot points, but I don't want you to get too worried about seeing how many they are. They are nice and easy to attack. If we have a look through, most of them, as we can see, are outline, describe, distinguish, one explain, where we'll need to look a little bit further, describe and solve problems. So even though there are quite a few dot points there, they will be quite easy for us to move through. So for all of you that have read or seen Harry Potter, you'll know that this here is Hermione. Hermione is what we call a witch. The big thing that we realise about Hermione from early on in the series is that she was born to two Muggle parents. So the interesting thing is that Muggle parents aren't actually able to carry out any magical powers. Now, how did Hermione end up being magical when her parents didn't have that trait themselves? So that's what we're going to have a look at today, is how traits are passed from one generation to another by going back and having a look at the beginning, at the father of genetics, a man named Gregor Mendel, and the experiments that he carried out. And then we're going to move forward and carry out a number of different genetic problems to see exactly how traits are carried from generation to generation. So in this lesson, we'll be mostly focusing on Mendelian genetics. So we'll start off by looking at who Mendel was himself, then looking specifically at some terminology that we need to know, and lastly, doing a number of Mendelian crosses, which will then use the information from the first two sections of this lesson in order to be able to carry out and solve a number of different genetic problems. So this is Gregor Mendel. He was an Augustinian monk who, during his time living in the monastery, worked on pea plants. And what he did was he looked at how different traits within these pea plants were carried from one generation to the next. So some of the traits that he looked at can be seen in this diagram here. So he had a look at how tall and short plants were carried from one generation to the next. He also looked at the pod shape, the pea shape, the colour of the peas, the colour of the pods, and the colours of the flowers. Now, it's interesting to note here that all of these characteristics are fairly easy to look at, and there's very distinguished differences between the two different traits. So a number of things led to Mendel's success when he was carrying out his experiments. Now, he chose easily identifiable characteristics. As we saw in the previous slide, there were definitely two main different traits for each of the different traits that he looked at. So obviously it's easy to tell a purple flower from a white flower or a yellow seed from a green seed. He used pea plants that were easy to grow. They had relatively short life cycles, so he was able to grow lots and lots of pea plants in a short amount of time. He began with what is known as pure breeding parent plants. So he took the time before beginning his actual experiments to make sure that the plants that he was using were only going to carry on particular traits. So these two particular ones here led to his experiment being extremely valid, which we know is important when it comes to first-hand investigations. The next thing was he used large numbers of pea plants. He didn't just do an experiment with 5 or 6 plants and think, yep, all good, we've got the right numbers, we're on track here. He actually used thousands upon thousands of pea plants. So as we know, another thing that we need to do in a first-hand experiment or first-hand investigation, is repetition, which leads to reliability. He also kept very accurate records. So while he was carrying out all of these experiments, he kept thousands of journals that had all of his results. His quantitative data was kept in immaculate condition. And then he was able to analyze this mathematically, which was quite an important thing to be able to prove that through his work, he was able to show that these traits were passed on in a particular way. So these obviously here fit with accuracy, which again we know is important when it comes to a first-hand investigation. And lastly, he controlled the pollination process. So by making sure that he and only he alone was transferring the pollen from one plant to the stigma of the other, was making sure that there was no cross-pollination happening, to make sure that the only thing that was working in this was our independent variable. So this one here also fits in with validity. Okay, so we've now had a look at how Mendel was successful with his experiments, but due to a number of different reasons, his work wasn't actually recognised for quite some time after he published his paper. So that was due to a number of different reasons. The first one was that he was a monk. So obviously, being within the religious sector, he wasn't really highly regarded by those in the scientific world. Secondly, he was Austrian, which meant that he spoke and wrote his papers in German. At the time, English and French were the two languages that most scientific communities worked in. So therefore, again, his paper wasn't really accessible to many of the people that were in the scientific world at the time. The next reason was his results didn't fit current understandings of inheritance. At the time, it was believed that the offspring actually got a blend of the different characteristics of both the mother and the father, rather than there being one or the other characteristic that could actually overshadow the other. And lastly, at the time, there was no understanding of genes or chromosomes. So scientists had very little understanding of the cell or what actually was inside the cell, and that didn't come along until the creation of more detailed or more specific microscopes that were able to look deeper within inside the cell. And therefore, when Mendel put this forward, this idea that there were factors within the cell that could cause inheritance to go one way or the other, people were extremely confused and didn't quite understand what he was talking about.